Learning objective 4 is about the basilar membrane and how it contributes to auditory sensation. Displacement along the basilar membrane is what results in our ability to hear sounds. Now, when we have sound waves that are traveling, there is a motion that occurs along the basilar membrane that begins at the base of the cochlea and then translates towards the apex. The pattern of that wave is different for different frequencies of sound, with high frequency sounds resulting in maximal motion of the basilar membrane at the base, and then lower frequency sounds result in maximal motion in the basilar membrane closer to the apex. This occurs because the basilar membrane varies in structure across its length, where at the membrane closest to the oval or the round windows, the membrane is quite narrow and stiff in structure, and the basilar membrane closest to the helicotrema is wider and more flexible. And those physical properties are what allow for the basilar membrane to have maximal motion at different lengths along the entire membrane structure. This is a graph that depicts the changes in basilar membrane motion along the different locations of the basilar membrane. So on the horizontal axis, this indicates the length of the basilar membrane, where nearest the oval window is down here at the left side of this horizontal axis, and then nearer the helicotrema or the apex is on the end of this horizontal axis. And it gives you the frequency of vibration at different regions along the basilar membrane. So you can see the high frequency vibrations occur nearest the oval window, and then lowest frequency vibrations occur nearest the helicotrema. And on the vertical axis, this shows you the width of the basilar membrane. So the width of the basilar membrane near the oval window is quite narrow. It is also narrow and quite stiff. And as you go along the basilar membrane toward the helicotrema, the width increases such that nearest the apex, the basilar membrane is much wider and therefore much more flexible. And as a result, high frequency sounds cause maximal motion or greater deflection of the basilar membrane where it is narrow and stiff, and the lower frequency sounds produce greater deflection of the basilar membrane where it is loose and flexible. Therefore, we can say that the frequency of sound, or the pitch, is coded by where along the basilar membrane there is the greatest deflection. Taking those properties of the basilar membrane into account, we can discern the types of sounds that are tuned by motion at the basilar membrane, given its location at the basilar membrane that is moving. So this is showing you an example of the basilar membrane that would normally be coiled up in the cochlea, and it has been straightened out and flat. And you can see nearest the base of the cochlea, it is narrow, and then nearest the helicotrema or the apex, it is quite wide. And this is showing you the sound ranges that will induce maximal motion at different lengths along the basilar membrane, with highest frequency sounds nearest the base, and then as you progress towards the apex, the frequency of sound decreases. Examples of high frequency sounds include very high-pitched noises, such as birdsong or a whistle. An example of low frequency sounds are low pitch sounds, such as thunder or a tuba. Learning objective 5 is about the cell signaling associated with auditory physiology. So unlike in the eye and unlike in the nose, the hair cells of the auditory system, they do in fact receive afferent and efferent input, but they're not neurons themselves. The hair cells are mechanoreceptors, such that, similar to the mechanoreceptors in the skin, they convert a signal, in this case a mechanical signal, into an electrical signal that is then detected by the afferent neuron that is nearby. Now, as I said, the hair cells themselves in the auditory system are not themselves neurons. They are actually polarized or a specialized epithelial cell. These polarized epithelial cells therefore have a basal or basolateral end and an apical end depicted at the top of the cell in the graphic and at the bottom of the cell down here in this graphic. There are stereocilia, which exist on the apical surface. They are stiff, graded in size, and rich in actin, which allows them to deflect or bend at their pivot points. And then down at the basal surface, there are synapses with afferent and efferent neurons at the basal end. And so therefore, this is where the mechanotransduction is converted into an electrical signal that results in the release of a vesicle containing a neurotransmitter or a neural signal that is then detected by the afferent nerve if it is the one that is innervated by, or vice versa, an efferent nerve fiber can release vesicles and signaling that would then be detected by the epithelial cell. In order to get into the cell signaling cascade that occurs when a sound is heard and then an electrical signal transduced, we have to look at the physiologic properties of the fluids that are surrounding and bathing these epithelial cells. First fluid to talk about is the endolymph. The endolymph is bathing the apical end of the hair cells where the stereocilia are located. This is a very potassium-rich fluid, meaning it has a very high concentration of potassium. And similar to intracellular fluid of other cell types, it's high in potassium, but it is low in sodium. Endolymph is located in the scala media, and the endolymph is produced by a structure called the stria vascularis, and we'll get into that later. The other fluid that is important to this process is the perilymph. The perilymph is the fluid that bathes the basal end of the cochlear hair cells, and it is opposite to the endolymph in that it is potassium poor. So you could say that this is similar to the extracellular fluid, such that it is low in concentration of potassium and high in concentration of sodium. The perilymph is found in the scala vestibuli and the scala tympani. This particular image is showing the kind of a big picture view of the signaling cascade that occurs following the transduction of sound, such that the basilar membrane is maximally displaced at a given location along its length that corresponds to the frequency of the sound that is moving it. Now, when the sound moves the basilar membrane is moving, and that generates a pressure differential that will then bend or pivot these stereocilia. In this case, the stereocilia pivot in the direction of the longest stereocilia. So they pivot in this particular image to the right. Now, when the stereocilia pivot or deflect, potassium ions enter the cell because these stereocilia are bathed in the endolymph. This is very high concentration of potassium. And when potassium is highly concentrated outside the cell, and then we have a bending of these stereocilia, potassium ion channels, aka these mechanically gated potassium channels, open, and then potassium will flow down its concentration gradient from the outside of the cell in this endolymph into the cell. And then whenever we start pumping in a bunch of, or at least flowing in a bunch of positive charges from the outside of the cell into the interior of the cell, the second thing happens. And that is we open calcium ion channels. And this particular calcium ion channel is a familiar channel called TRPA1. This is the exact same TRP receptor that we learned about in sensory and pain physiology. It's TRPA1. In this case, rather than being activated by something like mustard oil or something like that. This is a channel that's opened because of the change in voltage that is located here. Now, this ion channel opens, inflows a bunch of calcium, further adding to the positive charge on the inside of the cell. This depolarizes the cell. You can see down here the membrane potential goes up, and when that occurs, you have release of stored vesicles containing neurotransmitter. That neurotransmitter is then accumulated in the kind of the cleft or the synapse between the basal end of this epithelial cell and the afferent terminal of this primary afferent neuron, which then can activate this afferent neuron and induce a depolarization and potentially an action potential if there is sufficient depolarization occurring. So comparing a hair cell at rest to one that has been depolarized, you'll see even a hair cell at rest kind of maintains a constant partial depolarization, a very low state of electrical activity that just kind of spontaneously and kind of continuously is discharging. But during the displacement induced by the stereocilia, there is a bigger, more common and more frequent displacement and depolarization occurring, and therefore electrical signal. This is so sensitive that even vibrations in the basilar membrane as much as 0.3 nanometers, which is essentially the size of an atom, can cause channel opening in the hair cell. It's very exquisitely sensitive. So as we showed in the previous slide, deflection of the stereocilia that causes depolarization ultimately releases neurotransmitter. In this case, it is glutamate, and generation of action potentials in the afferent neuron that is located nearby. Now, should the deflection occur in the opposite direction, that is toward the shortest stereocilia, you get hyperpolarization, and that occurs because these potassium ion channels located in the tips of the stereocilia, they close. And when these close off, then those positive charges are no longer flowing in. The positive charges stay out in the endolymph, and then the cell can kind of revert back to its hyperpolarized state. When the hyperpolarized state occurs, then the voltage-gated calcium channels, in this case the TRPA1 channels, will close. Calcium is no longer flowing in, so the calcium ions stay outside of the cell, and then this can revert back to its negative or in fact a potentially hyperpolarized state. And then as you can see down here, there are no neurotransmitters being released, and then there is no activity in the afferent neuron, therefore no sound is heard. Learning Objective 6 is about the stria vascularis. Now this picture is depicting the cross-section of the cochlea with the organ of Corti in the middle, the scala vestibuli in the top, the scala media in the middle, and the scala tympani in the bottom. And along the lateral edge of the scala media is a barrier called the stria vascularis. That is essentially translated to vascular layer. So this is a highly vascularized portion of the scala media, this particular edge. And this is what produces the fluid that is going to be diffused across this barrier into the endolymph, and the endolymph then becomes high in potassium concentration. So we say that the stria vascularis maintains the electrical chemical properties of the endolymph, essentially keeping it very highly concentrated in potassium. So much so that it has an electrical potential of positive 80 millivolts, Whereas as you can see, the scala vestibuli and the scala tympani do not have that same high electrical chemical potential. The stria vascularis, as we said, is located on the lateral wall of the cochlear duct of that scala media. It produces the endolymph and it keeps it with a high level of potassium, around positive 80 millivolts. This high endocochlear potential drives the flow of those positively charged ions down their concentration gradient into the stereocilia and the interior cellular compartment of the hair cells. The stria vascularis forms what we call a blood labyrinth barrier, similar to the blood-brain barrier and other blood barriers in the body. This is a blood labyrinth, the cochlear labyrinth barrier. The BLB is one of the main sites of drug entry in order to access the inner hair cell. And this can be used for purposes needed to treat some sort of condition pharmacologically, but it can also induce some side effects. And any substance, whether it is pharmacological or something else, even carbon monoxide, that disrupts the function of the stria vascularis will diminish that endocochlear potential of that high positive 80 millivolts. And if that diminishes, then that can have an impact on hearing because there's less potassium to flow down its concentration gradient at the occurrence of hair cell deflection, or hair cell stereocilia deflection. And furthermore, the stria vascularis is a common source of autotoxic drug secretion into the cochlea. And autotoxic drugs can either target the stria vascularis itself and its fluid function, fluid diffusion function, or it can cross the BLB at this location and damage other components of the auditory system within the inner ear.